Okay, so today we are going to be talking about what happens when genetics gets weird. And our first example is incomplete dominance. And in incomplete dominance, it's not very clear cut. You don't necessarily have a dominant or a recessive trait. They're kind of neither. They tend to blend together, so you would see the red and the white color that could produce a pink color, and it would be called a heterozygous pink. So it says no allele is dominant or recessive. They blend together. So you're going to be using all capital letters. So a red flower would be denoted as big R, and the white flower would be big W. And then if you ended up with a pink hybrid, it would be big R, big W. So if we had the problem set up with two different pink flowers, notice we still are dealing with two big letters. So we have big R, big W for both parents. We put those on the top and the side of our boxes. Then we bring it down and over like this. Then we have to create the genotypes and phenotypes. Remember, the genotype is the actual genetics of it, whether or not it is homo or heterozygous, and then the phenotype is what that means. So with the genotypes, we had 1/4 of them that were big R. We had 2/4 of them that were big R, big W, and we had 1/4 of them that were big W. So what does that mean? That means that 1/4 of our flowers are going to be red, 2/4 of them are going to be pink, and 1/4 of them are going to be white. So our next example that we get into with stuff that gets weird is codominance. And this is where we get to talk about blood typing. So there are 4 different kinds of blood types that humans can have. You can have type A blood, type B blood, type AB blood, or type O blood. And there are 3 different types of alleles that are shown here. And here's where it gets super weird, because now we have superscript letters. So an I with an A above it, an I with a B above it, and then a lowercase I are the 3 alleles. So alleles IA and IB are co-dominant. Co means 2, and the allele little letter I is recessive. So both dominant alleles are apparent in the phenotype of the heterozygous offspring. And that's how we can end up with people that are AB blood. So all the possible genotypes for blood, there are quite a few. There are 2 ways that you could have type A blood. There are 2 ways that you could have type B blood. There's only 1 way that you could have type AB blood or type O blood. So our problem here says what types of offspring might be expected if one parent has type AB blood. So that would be an I with a big letter A and an I with a big letter B, with another parent that is heterozygous for type A. If the mom is heterozygous for type A, it would be big I, A, and then little I. So there we show the genotypes. We put one of the parents on the top of the boxes, one of the parents on the side. We bring them down and across, and then we get to our analysis. The genotype is just the genetics of it. What letters are present? 1/4 of them are IA. 1/4 of them are IA, IB, and 1/4 of them are IA with a little I, and then the last 1/4 is IB with little I. So then you have to say, well, what does that mean for the phenotypes? So notice there are 2 types that would end up in type A blood. The 2 big A's would give you blood and type A blood, and you would also get type A blood from IA little I. You would have 1/4 possibility of having type AB blood because you do have the IA and IB. And then the only other possibility that you could have with IB and the little I is that that person would have type B blood. So our last example today of when stuff gets weird is with multiple alleles. And you usually only hear of lower animals being talked about with this, such as rabbits or chickens. You usually don't hear human examples. So they have many genes that are expressing one allele, especially when it's called multiple. Color code in rabbit is determined by 4 different alleles. So you would have the capital letter C that would be the full color. You would have a little c with a subscript CH that would be a light gray or what they call a chinchilla color. You would have a lowercase c with a subscript H that would be albino with black extremities, which is what they call a Himalayan. And then you would have just the lowercase c that would be the albino, just completely white rabbit. So there are 4 different possibilities for that one type of rabbit. So then each one of those have possible genotypes or the genetics that you could see. You see all of those listed for full color, all of them listed for chinchilla, all of them listed for Himalayan, and there's only one option, very recessive, very unlikely, that you would have a pure white rabbit. Then when you get to a problem, this one says, what types of offspring could be produced by a full-colored rabbit that has this genotype that's bred with a Himalayan? You put one parent over top of the boxes, one parent on the sides, and fill them in like this. And then you have to look back at the genotypes and the phenotypes. The genotypes are just the genetics, so you just pull those directly out of the boxes and put them in the chart. You will have 1/4 of them that will be a capital C with the little c, H. You will have 1/4 of them that will be the little c with the CH and the little c with the H. You have 1/4 of them that will be capital C, little c, and then 1/4 of them that will be little c with a CH and little c. For your phenotypes, you have to look back at that last slide to figure out which ones of those would be full color. And the ones that have the big letter or capital letter C would show the full color. And then the other 2, because they have the C with the subscript or superscript CH, those would be the